Good afternoon. Myself Rudra Gouda, a PhD student from Division of Entomology. I welcome you all to my merit medal presentation. As we know that crops are severely affected with the pest. The one such pest is whitefly Bemsa tabaci, which is highly polyphagous in nature, having the host range of more than 600 plus plant species, and it also transmits many plant viruses. And in this Bemsa tabaci, there exists a cryptic species complex. Around total 46 cryptic species complex are present in the world, of which 13 are present in India. Among that, Bemsa tabaci Asia 2 found to be a predominant one. And in recent past, in Punjab and Haryana, this pest caused an epidemic in the cotton crop and a loss of around 4,200 crore rupees. And this pest has gained resistance and resurgence to almost all the insecticide, and as a result, there is a need for eco-friendly alternative to develop. One such eco-friendly alternative mechanism to target is olfaction. Insect normally uses a sense of smell to recognize a host, mate, and oviposition site identification. Here rises a question: how the insect finds the host, and what are the genes involved in this, and how many of these genes are present, and what are their function, whether we can use these genes for sustainable management of whitefly. So these are the 4 major proteins involved in the insect olfaction, of which chemosensory protein and odorant binding protein are important and are dealt in this study. When coming to research gap, only few of these genes have been identified in Miam and Med-Genti group, but information of these genes is absent in the Asiatic Genti group, which is predominant in Indian subcontinent, and also how these genes govern the function of the whitefly is also very less known. So based on that, we have designed this topic, that is genomoid analysis and identification, characterization, expression and functional analysis of odorant binding proteins and chemosensory proteins in the whitefly Bemsa tabaci. Coming to objectives, my first objective is identification and characterization of OBPs and CSPs from Bemsa tabaci genetic group, and objective 2, expression analysis of select OBPs and CSPs in Bemsa tabaci, and objective 3, functional characterization of select OBPs and CSPs from Bemsa tabaci. So when coming to objective 1, we have taken almost all the OBP sequence identified in insect till now, and we blasted in the genome of the Bemsa tabaci across the different database, and we got the hits for those genes by using a T-blasting tool with a e-value cutoff of 10 to the power of -5, and further these genes were validated by designing the primers. So we collected all the life stages of the whitefly separately and extracted their DNA, and our desired PCR primers were amplified, and Sanger sequencing was performed, and Opten sequence were submitted to NCBI, and we used a robust computation pipeline where we around 35 bioinformatics tools were used to perform a downstream analysis, and this is the first genomic analysis in OBPs and CSP genes in Bemsa tabaci Asia 2 and we identified 14 novel OBP genes. Among them, 6 were novel and 14 CSPs in Bemsa tabaci Asia 2 and we also found another group of important protein called as ejaculatory bulb proteins. For the first time we identified the ejaculatory bulb proteins in insects, and these are nothing but a homologous to that of the chemosensory protein, but they are highly restricted to the male ejaculatory bulbs. And also we first time gave the complete gene sequence of all these genes in the Bemsa tabaci Asia 2. So here comes the question: what are their structural and functional features, and how they are localized on the chromosome, and what are their intron and exon details. So all these genes identified have a single exon structure except CSP 1, 2, 3, 8 and 14 having an intron in them, and chromosome mapping shows that majority of these genes were congregated on chromosome 3, 4 and 6. So this indicates there is a clustering and non-random distribution of these genes, and these genes may express the influence of expression of the other genes at the same locus. And further we identify the retrotransposon insertions, and in the genes, those genes which is having intron have the retrotransposons. Among our identified CSPs, CSP 1, 2 and 3 were found to have a retrotransposon insertions. These retrotransposons are nothing but genetic elements that can move in a genome via copy and paste mechanism, and these retrotransposons helps in creating many functional proteins by using a RNA splicing technology. And in order to prove any protein it has a chemosensory protein, it should have a 4 conserved cysteine residues. So our identified all the CSPs have a 4 conserved cysteine residues, and similarly ejaculatory bulb proteins are also a type of a chemosensory protein, and they too have a 4 conserved cysteine residues. And when coming to identifying of OBPs, OBP protein should have a 6 cysteine residues, but this is not a rule and there may be exception. Based on the number of cysteine residues, it has been classified as classical, dimer, plus C, minus C and atypical OBPs. So accordingly our identified OBPs have been grouped into these classes, and we further regrouped all the OBPs identified in the whitefly. Previously they were wrongly classified as classical OBPs. So this is our updated classification of the OBPs based on the cysteine residues, and we have published that in our International Journal of Biological Macromolecules. And we also performed the phylogenetic analysis by taking all the OBP sequence identified till now, and we found that OBP8 were found to be ancestor for Bemisia Asia 2 1 OBPs, and similarly CSP4 was found to be ancestor for other chemosensory proteins in the whitefly. And coming to silent findings of objective 1, this is the first genomoid analysis in Bemisia Asia 2 1, where we identified 14 OBPs, among them 6 were novel to the science, and 14 CSPs and 10 novel EBP. We gave complete gene sequence of all these genes, and retrotransposon insertions and ancestral gene identification have been done. Now comes the question whether the expression of these genes is vary across the tissues and across the life stages of the insect. For that, we have performed the functional expression analysis, where we have checked the expression of all these genes in the developmental stages of an insect versus adult, like nymphal versus adult, and across all the body tissues like head, thorax, abdomen, leg, and wing. And we found that in the differential expression, OBP8 were highly expressed across all the body tissues and across all the nymphal stages. So it play a very vital role in the life cycle of the Bemisia tabaci, and most of the OBPs and CSPs were highly expressed in the adults, so they may be involved in the host recognition, feeding, and reproduction, whereas CSPs were highly expressed in third and second instar, so they may be involved in the immune response. So further spatial expression across the different body parts like head, thorax, abdomen, leg, and wing shows that OBPs were highly expressed across all the body parts, and CSPs also followed the similar trend, with the CSP9 and 10 were down regulated. And ejaculatory bulb proteins, these are especially found only in the male reproductive tissue, and the expression of this gene were high in the male before mating, but after mating in the male the expression of these genes was reduced, but in female before mating the transcript of these genes were not found, but after mating transcript of these genes in the female was found. So this gives a clue that EBP might have transmitted from male to female during the mating. So coming to objective 2 findings, QPCR demonstrated that spatial and temporal distribution of these genes in Bemisia tabaci, and also EBP might be transmitted from male to female during mating. So here comes the question, now which of these genes we need to take for functional characterization, and whether these genes play a role in host recognition or insecticide resistance, or whether these EBPs are really transmitted from male to female during mating. For that, we have conducted the functional characterization, where we have followed the 3 different workflow for OBPs, CSPs, and EBPs separately, which I will explain in coming slide. For first, when coming to OBP characterization, for first we performed olfactometer bioassay, where we found that beta caryophyllene and carvacrol were having attractant activity for whitefly, and beta asimine and repellent activity in the whitefly. And OE position inhibition assay shows that alpha pinene and beta inine and beta asimine are significant OE position inhibitor at a lower concentration of 0.05%, and similarly the beta caryophyllene and carvacrol are OE position inhibitor at a higher concentration. So further, we selected OBP3 and 10 because they were highly expressing across all the body parts and also body tissues. We have taken further for construction of a protein structure for in silico docking with the volatile organic compound, which are shown good attractant and repellence in the whitefly in the bioassay. For when coming to in silico docking, we found that beta inine and beta caryophyllene and beta pinene having a high binding energy, and there are still more volatile organic compounds which are showed high binding energy, but they are not disclosed because of a scope of product development. And we also cloned the 2 proteins OBP3 and 10 in expression vector BL21DE3, and we further extracted the pure proteins of OBP3 and 10, and this is a gel picture of SDS page, and we got a OBP3 protein of molecular weight of 35 kilodalton and OBP10 of 32 kilodalton. And further, competitive fluorescent binding assay with the purified protein has been done with the BISANS as a fluorescent reporter in order to validate the findings of the in silico docking, and we found that beta inine, beta caryophyllene, and beta pinene were having high binding energy similar to that of the in silico docking. So further validation was done by silencing these genes by RNAi. Both OBP3 and OBP10 was silenced, and the silenced population when we subjected to olfactometer assay previously, which are showing attraction to caryophyllene and carvacrol, but now they show the repellence activity. So this confirms that OBP3 and 10 are involved in the host recognition. And output of this OBP study, the functional validation proves that OBP3 and 10 involved in host recognition and OE positions identification, and caryophyllene and carvacrol acts as attractant and asimine as a repellent, and alpha pinene, beta pinene, and beta asimine are a significant OE position inhibitor at a lower concentration. So further, for functional validation of chemosensory proteins, we perform the transcriptomics before and after exposure of insecticide, and which are the chemosensory proteins which are highly expressing. They were further validated by qPCR technique by treatment with imidacloprid and fipronil. After 6-hour treatment, we found that CSP1 and CSP5 are highly upregulated for both insecticides. So they were further perform in silico docking by constructing the protein structure and docked with fipronil and imidacloprid, which are having very high binding energy, and then silencing of these two genes were performed, and after silencing CSP1 and 5 show 45 to 50% more mortality. So, for in function of validation of EBP genes, EBPs were highly expressed in the male before mating, but when it was silenced, the transcript of these EBPs were absent, and also in male and female. So, this indicates that EBPs were clearly transferred from male to female during mating. Implication based on this is the first study, so it gives a new target for genes and mechanism to target. Based on this OBP3 and 10, we can disrupt for pest reproduction, and this attractant repellent can be used for developing eco-friendly pest control methods, and these oviposition inhibitors can be used to reduce the pest population. So, we can develop insect management resistance strategies based on this information, and the transferring of EBP concludes that we can go for mating disruption in male and female. And based on the leads obtained in this study, we have developed the lures and performed a multi-location trials, and we found very good results. And there is a chance of product development around 4 to 5 fold more insect trapped when compared to that of control. Future direction, we need to characterize the remaining genes by these techniques, and by this we can develop a suitable control strategies. These are 5 publications with NOS rating of 14.20 and NOS with 10.60, another NOS 10.60, and another 2 paper with 8.80 and 6.70 NOS, and these are 2 papers are under review, and these are the nucleotide and protein sequence generated. These are the NCBA bio project. These are IARI database created, and we have granted technology from ICR, and bioinformatics pipeline developed in this study. These are best orals and poster in national and international award, and apart from that, these are 5 awards, and I also served as a student associate in Indian Journal of Entomology, and I got I was served as a course associate and trainer in NIAB caste and DST training programs. These are trainings attended. This is advisory committee, and acknowledge ICR IARI, Dean, Director, Joint Director, Graduate School, Chairman, and Advisory Committee, Head, Division, and Professor of Entomology. Thank you.
Rudra, this is an exceptional work. I must congratulate you and your chairman for doing such a marvelous work.
Thank you, sir. Thank you, sir.
You name it, you have done everything possible by addressing different research questions through a very highly complex area. Oh God, how much work you have done, man.
Thank you, sir.
This is truly,
Thank you, sir.
truly brilliant, and you have published such high quality articles, and it's really marvelous to hear IRI student doing so beautifully.
Thank you, sir.
Keep it up.
Thank you, sir.